Hi guys. So we're going to talk about how this plant here can get a light snack. And essentially that is the process of photosynthesis. We're going to do a bit on an introduction to photosynthesis and talk about what light energy is. And then we're going to actually get into the biochemistry of photosynthesis by talking about basically how it works both with light and then how it essentially takes the products that light gets turned into or transferred into to actually make the sugar molecule. So let's start now with an introduction to photosynthesis. So we talked about last week, or in our last unit actually, about cellular metabolism. And cellular metabolism is a way for which we can take sugar molecules and essentially break them down, or other molecules. We're just going to focus on sugar or glucose specifically now, to make the little molecule adenosine triphosphate, or ATP. And essentially we use that little molecule ATP to essentially allow tons of reactions to happen in our body, or for physiological changes like being able to have muscle contractions happen so we can walk and run and pick up our pen. Well, essentially we can start asking, where does that sugar come from? And that sugar, in many times, comes from plant sources. And plant sources are able to get that sugar by making it. And they make it through a process called photosynthesis. And photosynthesis is essentially just being able to capture light energy and convert it into chemical energy and or to make sugar. So the particular things that make these sugar molecules themselves are called self-feeders, or called autotrophs. So these are things like plants. Right? You can get certain sea slugs that essentially have the capability of doing photosynthesis, or even things like cyanobacteria. Right? Can essentially be self-feeders. Now, heterotrophs are things that obtain nutrients by ingesting other organisms. So animals are a perfect example of these, right? So we're going to focus on how autotrophs essentially make their food through photosynthesis. So photosynthesis actually has two reactions. They have the first one, which is the light-dependent reaction. So if you can try to figure out when this happens, during day or during the night, you would be right, during the day when there is light. Right? And so we use things like pigment molecules to absorb the light, and then electron carriers and ATP synthesis to move the energy through enzymes to essentially make one molecule. You're going to see ATP. And we're going to show where these are located in the stroma and the thylakoid membranes. Now, the light-independent reaction, which is also known as the Calvin cycle, can happen during the day or night, right? It used to also be called the dark cycle. I like light-independent because dark cycle, you might think that it just happens at night, but it doesn't. Light-independent means it doesn't need the light to happen, but it can happen at any time. So these are the two reactions that we're going to focus on today. So essentially how this works, let's start with what we did before. Essentially, we were talking about having ATP, or metabolism making ATP. Well, the light-dependent reaction works much the same way. But you can see what you're going to find out, that photosynthesis, when we look at the overall equation, is going to look a lot like cellular metabolism. The difference is here, if you see, we're bringing in water and essentially light energy. And then what we're making, like bring in right here, right? And then what we're making here, essentially, is ATP and this thing called NADPH. And what you'll notice here, there's a P in it versus NADH that we talked about with metabolism. That one, I was able to remember it because it's for photosynthesis. And oxygen is a waste product in this reaction. And then we take this ATP and NADPH and we shuttle it into the light-independent reaction. We bring in carbon dioxide from the outside, right? Remember, these are plants that are generally doing this. And then we make sugar here. And essentially, we make this sugar by taking off some of the energy here, like doing that hydrolysis reaction, kind of cutting it down, right? And then basically taking some of the electrons here. So you can see we get this to start the reaction. So this reaction essentially is, in a way, cyclic. So, true and false. Does a light-independent reaction produce NADPH and ATP? This would be false, right? It would be the light-dependent reaction that actually made these two guys. So let's talk about where photosynthesis happens. Now, this is a plant, and we've done this cross-section of a plant here. And you can see right here, these are the epidermal cells on the outside. And in this middle area is what's known as a mesophile. And this is where we're going to see the root of cellular respiration happen. Now within here, you can see there are photosynthetic cells. And we're going to pull one of those out and look at it a little bit closer. Now, the one thing I want you to pay attention to is there are these things called stomatas here. And as you can see, they open up and allow for the flow of gases. So they allow oxygen to leave and carbon dioxide to enter, right? And so that's going to help regulate some of this. Now, if you look at this individual plant cell, and we looked at these a couple weeks ago underneath the microscope, you'll notice these guys right here. And that's where we're going to start seeing what's known as chlorophyll, but those are the organelle known as chloroplast. Now, the chloroplast, if you kind of make it a little bit bigger, you'll notice that it has a double membrane here, just like the mitochondria did. This is the outer, this is the inner. Now, the place where everything's just kind of open there, that essentially is what's known as the stroma. The stroma, I like to think, is a lot like the mitochondrial matrix, if you're comparing them, right? And then you can see here that these little things that to me look like York peppermint, green York peppermint patties. Essentially, a stack of these is what's known as a grana or granum, right? So if you see a grana, stack it. And these individual peppermint patties are what are known as thylakoids, right? So a stack of thylakoids is a granum. And the thylakoid, in it, you'll notice the inside here. Actually, let me change your color really quick. If you notice right here in this middle part, that is a lumen. And the outside of it here is the membrane. And the lamella, are those that connect the two granums. So this is going to allow different things to happen. For instance, when we start talking about the light dependent reaction, that's going to happen in this area because that's where the enzymes are going to be basically stacked together. And when we start talking about looking at the Calvin cycle or the light independent, that's going to be basically happening in the stroma area. So let's find those that apply here. So site of photosynthesis, choose all that apply. So are chloroplasts mostly found in cells in the epidermis of the leaf? No, that is not right, right? Chloroplasts, do they have several compartments? They do, don't they? You have the thylakoids, and within the thylakoids, they're stacked up to granum, so you do have, it has been compartmentalized. Our thylakoids are membranous components in chloroplast. They are. The matrix is a liquid component of the chloroplast. That would be wrong, right? Like I mentioned, matrix is in the mitochondria, and that is the area around the thylakoids. The area around the thylakoids is the stroma. And the last, the grana is a stack of thylakoids. That is correct as well. So photosynthesis, essentially, if you look at the equation, you're going to start with 6 carbon dioxide molecules, 12 water with light in the pigments, right? And that's going to equal a sugar, 6 waters, and 6 oxygens. Now, if you look at the overall equation here, this looks just like cellular respiration. The difference is it's backwards. And you might be going, wait a minute, well, there's 12. Well, if you take 12 here and 6 here, that would make this 6. So technically, right, plus energy goes to sugar plus 6 CO2, right? Or I mean, sorry about that. Let me go back. Let me fix that. Sorry. 6 oxygens, right? So essentially, if you flip this backwards, this is what cellular respiration looks like. So the great thing about this, guys, is if you remember the equation for cellular respiration, then you just have to remember if you do it backwards, it's photosynthesis. So we are going to look at how all of this works. So let's talk about light, because that's what essentially starts everything, right? Now, light, otherwise known as electromagnetic radiation, is a form of energy. And essentially, these guys travel in waves. So when you have light that comes through, it essentially is going back and forth and traveling in this wave. Now, from one top to the next top, that's what's known as a wavelength. So depending on how much energy you have, this can get longer, wider, or shorter. Now the crests are the tops and the troughs are the bottom, right? And when you look at the change from how tall they are from here to here, that's what's known as amplitude. So one cycle you can see goes from where it's about to start at the line to the end. So this is one complete cycle. Most of the time what we do is measure from wavelength from crest to crest. Now, you can see here, this is the electromagnetic spectrum, and this is looking at these wavelengths. So do you see how these are wider across? So these waves are bigger here, and essentially this has less energy over here. Bigger waves have less energy. Over here, if you look, these guys, the wavelengths are really, really small. And the small ones have more energy. And I kind of think of the idea of how this kind of plays together is if you essentially have a ribbon in your hand and you're basically flowing your hand up and down and making the ribbon go and kind of flow up and down like this, The more energy, to make it look like this, would require you to flip your hand back and forth a lot faster and use more energy. Where if you wanted it to look longer, you could basically wave your hand up and down less. So that's how I remembered the difference about what had more energy and not. So things like gamma rays, X-rays, ultraviolet, this is on the more energy, the smaller side. Visible light is what we're going to focus on here. That's essentially what we can see, and you'll think Roy G. Bivs, right? And that goes from basically less energy to more energy if you look, because you can see where the red starts here and it goes to the blue. Red in Roy G. Bivs basically stands for Roy G. Bivs, and that should help with red, orange, yellow, green, blue, indigo, and violet. And like I said, this has less energy on this side, so these waves are bigger. This is more energy, and so the waves are smaller. Okay, so that's the only electromagnetic radiation we can see. Okay. So the range that we can see, if you notice, is between 380 nanometers to 750 nanometers. And I don't have it on there. But that's the actual range or the wavelength that we can actually see with our eyes. And it's kind of funny. Photons, they're fixed amounts of energy. And what's funny is though they travel like a wave, they can interact with particles, or they can interact with things like their particles, or they can collide with them. So they kind of have both properties. And just remember, the amount of energy is inversely related to its wave. So the larger the wave, the less the energy. The smaller the wave, more energy, right? And so this is important because how we're going to essentially make sugar is our sugar is a way of holding energy for our cells, because essentially the whole purpose of everything for you to make ATP is so that you can move energy around in a usable form in your body so that you can give the energy to reactions or mechanisms to happen. And so essentially what we're doing is we're taking the energy from the sun, or the plants are, and they're putting it into storage molecules. So how we do that is essentially we have pigments that will absorb energy. And the color of the pigments essentially work in the way that if you see a particular color, it's because it's not reflecting that wavelength or that color. It's bouncing off and your eye is detecting it and your eye is absorbing it. And so it'll absorb all the other wavelengths besides this, right? So the biggest, plants like to absorb violet colors, which is essentially blue and red light. So if they absorb the color, the light disappears. If they don't absorb it, then it's going to bounce back and reflect to you. So for instance, when you're talking about something being black, essentially what's happening there is they're absorbing all wavelength. So you're seeing that color because the absence of any wavelength being reflected back to your eye at that time. That's a reason why if you want to stay warm, you go outside in a black sweater, because then essentially all the light that's coming down, all that energy will be absorbed by that outfit, and then essentially you can stay nice and warm. Now, if something is white, this is why on hot summer days they tell you to wear white, because it reflects all wavelengths. So when you look at something that's white, that's essentially every wavelength coming back and being absorbed into your eye and reflecting off of the paper. So that's why essentially you see those different colors. So this is what's known as an action spectrum. And these are basically how different pigments within leaves or plants work. Now we see the color green from plants, sorry, because essentially that's chlorophyll is the pigment. And so chlorophyll is going to, if you notice, there's chlorophyll A here and chlorophyll B. So we do have different pigment slightly, and the color you see is essentially based on what's being reflected back to you. If you know here, this is chlorophyll B. So do you notice how it goes down here? It's not absorbing because this is how much light we're absorbing, and this is the wavelength of light. And so they're kind of giving you here, right, that you have red over here. And you can see that's 700. So these are large wavelengths here, right? Less energy. This is more energy and shorter. That's why these numbers are smaller over here. But if you notice at the sky, right, it's not absorbing at all in the green region. That's essentially why you see chlorophyll B as being green, right? If we go and we look over here, the other one we have is this, right? And this is the other chlorophyll. And as you notice, this one also is not absorbing at all in the green. So they're gonna, because they absorb slightly different ranges, they're gonna be slightly different colors of green. One's gonna be a little bit more yellow than the other, right? This one, essentially, if you see here, absorbs a little bit more in this like yellowy region, right? So this one's gonna be a little bit darker than this one. Sorry. So essentially here, this is because they don't essentially at all absorb anywhere in the green. That's why you're going to see these two green. Now, if you notice here, though, not everything is green. There's another line. Let me change colors again. And this is the carotenoids. And so this is the carotenoid right here. And you notice from here on, there's no absorption, right? They absorb all down here. Now this, if you think carotenoids, it may sound like something that you have heard of before, like beta carotene. Now, where do you find beta carotene? Hopefully you said carrots. And that should kind of give you an idea. Carrots are that orangey color. And it's because, as you can see here, he is not absorbing at all in this area. So this is what you're going to see. And so you get a mixture of a little bit of green all the way through the red. So you're going to get that kind of orange feature. But he's absorbing down here. So if you look at this, this should give you a good idea of what color gets absorbed the most. And if you were trying to be the most efficient you possibly could be, you would give plants blue light for the most part. Because most of what we look at when we look at different types of pigments that are mixed in plants, they all essentially absorb in blue. Now, you may not realize this, but think, we live in the South, so we don't really get to see leaves very often change color. But if you go up to the north, you'll just see it's absolutely beautiful. And as we get into fall, this time of year that we're in, right? Or once you get into fall, what you'll essentially see is you'll see the leaves start to change colors, right? They'll go from green, and then you'll start seeing where they're red and they're yellow and they're orange and they're brown and all these different colors. And essentially what is happening there is the chlorophyll molecule is really not very stable. And so as soon as the trees start to basically try to protect itself for the winter, so it'll cut up all the nutrients to the leaves themselves. And so that's why you start seeing all the leaves fall off. Well, they don't just automatically fall off. It takes a little bit of time for those leaves to die. And so in the process, once it stops getting nutrients, then the chlorophyll pigments will start degrading. And that's when you'll start seeing the other colors, because those color pigments are more robust, so they won't degrade. So that's what you're seeing. So when you're looking at a leaf, a lot of times there are a lot of these different pigments there to try to capture as much light energy as possible. We just tend to focus on chlorophyll because it's the one that's most plentiful. So which light would plants grow best exposed to? Yeah, it would be this blue light. And a lot of places have started doing this. It started out in Japan, where they made these huge, tall buildings. And we're doing it now here in certain locations in the US, up north a lot. So what they'll do is they'll make these high-rise buildings. And within them, they'll grow floor upon floor upon floor of different plants. And so what you would do is, they have everything kind of like a greenhouse of some sort in there, where they can control temperature, they can control the amount of water, and they essentially would put blue light on them versus white light that you might see in your office or in this class or in your house or in a classroom. There's a lot of wasted wavelengths being used there. So if you just generate blue light, then essentially they have a higher percentage of light that they are essentially absorbing, and you're wasting less energy. Now, what color do you think those plants look like under blue light? If you didn't guess it, they would be black. Why? Because they're absorbing all the blue light, so you wouldn't see anything. You take that same plant outside, though, and it's going to look normal, like you would expect it to. Now, plants appear green because they absorb green light. True or false? False. They appear green because they reflect green light, not because they absorb it. If they absorbed it, you wouldn't see it because it couldn't come to your eye. So I'm going to talk a little bit about this next section, and this is where we're going to start next time when we talk and kind of review this a little bit again. So essentially what's happening is we are taking in light energy in plants, and then we're going to move that energy through enzymes by using electrons or to do them. So essentially the energy will go into electrons and excite them and make them go into high energy. And if you remember, basically what happens here is electrons move from high energy to low energy, just like diffusion happens, right? With concentrations or molecules. Everything likes to naturally move from high to low. So let me show you what we're talking about. So we're going to start with what's known as light-dependent reaction. We're going to have pigment molecules that are going to absorb light. That light or that energy is going to be transferred with electrons, and they're going to go through two reaction centers. Those reaction centers are going to be called photosystems. And that's going to allow us to basically capture the energy in electrons and move them and actually put that energy into molecules known as ATP and NADPH. So, the light energy happens in the thylakoid, and we convert that energy into ATP and NADPH. And remember, don't get it confused with NADH because this has P for photosynthesis, right? And so we have what's known as cyclic and non-cyclic and cyclic electron flow. And so let me show you really quick what cyclic is. I mean non-cyclic. So your eyes are going, oh my goodness. But it's not as hard as you think it's going to be. So just hang in there for a few minutes and essentially we will get through this, and this is the last part I'm going to talk about. So essentially, if you notice this, remember how I was talking earlier about the thylakoids? Those, they look like basically peppermint patties on top of each other, and they were green. Those are the peppermint patties. That's the lumen part that's on the inside, and this is the stroma. So just like when we had cellular metabolism and the electron transport, if you remember, we had enzymes in the membrane. And what these enzymes did is they just pushed electrons through them. The electrons were carrying energy. Okay? So that's all it is. So electrons carrying energy through these different enzymes. And they're doing it basically to put that energy onto, or put those electrons onto NADPH. Okay? Now, what do they start with? Well, there's a few things we need. So you see here, if you remember cellular metabolism, we basically started with oxygen. That's why when you work out, you start breathing so much, because you need oxygen to your muscles to make more ATP. Well, because remember how photosynthesis is opposite everything. Essentially what you're going to see here is that you're going to take water, you're going to split water and then release oxygen, right? And so we split water using this, which is called the water splitting complex. And that is a protein that's attached to this guy, and the whole thing is called photosystem 2. Now, photosystem 2 is first. If you notice here, there's another one called photosystem 1. This one's second. Why? You're like, why do they have to be so confusing? Because they named them in the order they were discovered, not the order that we see them. And so it's harder later to go, okay, let's rename these in the order that we use them. No, people have already made a foundation and have all these papers published. So essentially they just called this 1, this 2. And so we always start with photosystem 2. So essentially we have water. We split it in half. The oxygens go one way. The hydrogens go another, and they're going to get essentially moved over here. And then we're going to steal the electrons to start pushing them through here. Well, the electrons are here, and they don't have a lot of energy, right? Electrons don't have enough energy to move. It's kind of like us. We're on the couch. We don't have enough energy to move. So let's get energy. So they bring in light energy so that they can move. Right? So that's what this whole process of photosystem 1 is. So we split water, take the electrons from the water, move them into photosystem 1, and then all these chlorophyll molecules are going to basically take up energy, right? They're going to, much like a gutter does with water, they're going to essentially move the energy down, bring it to this area over here, right? Give it to the electron, and then the electron's going to go, woo, and move through the enzymes. Okay. As we do this, you'll notice things like hydrogen start pumping to one side, right? That's important because if you remember last time that we talked about cellular respiration, we needed hydrogens to activate ATP synthase, right? So basically this whole time, the electrons are going to move through here and then they're going to dump on NADP plus to make NADPH, okay? So that's what the whole process here is doing. And then of course, if you look at enzymes, they tell you what they do. NADP plus reductase is reducing NADP plus. And if you remember, Leo says GR, loss of electrons, oxidation, gain of electrons is reduction. So they're basically adding electrons onto NADPH and gaining electrons. So that's how you know it's a reduction reaction. Now, so the electron, the movement of electrons through make NADPH. Now, as this happens, as I told you previously, you have hydrogens coming across the membrane here, right, in this area. As hydrogens build up, do you see there's so many of them? It's going to change the charge, but more so, it's going to turn on this enzyme ATP synthase. And so essentially these guys are going to go down here much like the water will, and then what's going to happen is we're going to take NAD plus to make ATP. So essentially we make NADPH and ATP here to go to the next reaction, which is a light independent reaction or the Calvin cycle. So if we need both these molecules, then we go through, this is non-cyclic. And can you see why it's non-cyclic? It's not going in a circle. It essentially goes and starts, if you notice, starts over here, goes through, and comes out here. Nothing goes back on itself. Now, if you just need ATP, if you just need ATP, then you do cyclic. And essentially, this is just with photosystem 1. And so you get it coming through here. This is where that hydrogen went through. And the electrons come here. But instead of moving on and continuing to NADP to make NADP+, there's not enough energy, so it goes back. And so it goes around and around and around like this. So that you can continue with the hydrogens pumping across. And so this is sometimes when we need just ATP. Realize ATP is used for so many different reactions. We're using it all over to make other enzymes work and just to allow the overall function of the cell to happen. So there are going to be times that you need ATP more than NADPH. So that's the reason why this happens. So cyclic uses both photosystem 2 and photosystem 1 to move electrons through to make NADPH, and the movement of electrons makes hydrogens go across to make ATP. Those get moved into the next cycle, which we're going to talk about in the next video or face-to-face. If you just need ATP, what happens then, I'll show it to you on here, this is non-cyclic. This is cyclic, sorry. Cyclic. And so it comes through here, and instead of going on here, it doesn't do that, it goes back. Just so that you can get more hydrogens pumping across, so you can make just ATP. So with that being said, I'm going to go through these questions later when we are actually in class. So that's all I'm going to talk about for this video, guys. And looking forward to seeing you later.